In Chapter 9, we're going to explore the different types of catalytic strategies that are employed by different enzymes. So just to look at an outline of the overall chapter, we'll look at the 4 classes of enzymes and their catalytic strategies. So when we're talking about the classes of enzymes, we're talking about just examples of some enzymes that are present. So we'll look at things like serine proteases, carbonic anhydrases, restriction endonucleases, and myosins. Now, in studying these, we do need to look at the different types of strategies that exist. So we'll first talk about binding energy, but the actual types of catalysts, the actual types of catalysis include, there are 4 different types: covalent catalysis, general acid-base catalysis, catalysis by approximation, and metal ion catalysis. So we'll define and explore each of these different types, and we'll look at examples of the different strategies that exist. So in our study of catalysts and enzymes in other courses and in the past, we're introduced to the idea that enzymes are going to catalyze reactions and do so by stabilizing the transition state. Okay, so in all of these, the goal is to stabilize that transition state. And what this does is it lowers the activation energy, and lowering the activation energy allows us to speed up the reaction. So in doing so, the binding energy actually allows the reaction to occur at a faster rate. So the binding energy is involved in this process of catalytic conversion of substances. So it's important to keep in mind that binding energy because we're trying to interact the substrate and the enzyme and do so in a position where we're lowering that energy that it takes to do so. But again, the actual types of catalytic strategies that we need to define are these 4 right here: covalent catalysis, general acid-base, catalysis by approximation, and metal ion catalysis. So starting with covalent catalysis, many enzymes actually will contain active sites that have catalytic residues that form temporary covalent bonds. So temporary covalent bonds that are forming in these active sites. And they form these temporary covalent bonds with the substrate molecule. At the end of the reaction, the covalent bond has to be broken because we have to be able to release the substrate and then regenerate the enzymes. That's why these are temporary covalent bonds that form. With acid-base catalysis, active sites may contain residues like histidine, but the residues can participate in hydrogen ion transfer. And we know that proton transfer is what's involved in acid-base reactions. So by transferring this proton, the active site may activate nucleophiles that are required in catalysis. So it results in activation of nucleophiles, and those nucleophiles would be required in catalysis. It can stabilize charge groups. And it can increase electric interactions that stabilize the transition state. So increase interactions to stabilize transition state. With catalysis by approximation, many biological reactions will involve 2 or more substrate molecules. So this implies that for the reaction to occur, these substrate molecules must be close enough and must also have a proper orientation in order to interact. So This particular type, this catalysis by approximation, is going to rely heavily on collision theory. And if you remember from general chemistry, when we talked about collision theory, we talk about how the molecules must have enough energy in order to interact. You have to have a high enough concentration in order to interact. They have to be in the proper orientation in order to interact. So molecules have to collide in order to be able to form products. Okay, so they have to collide with enough energy, with proper orientation, and it's the microenvironment within the enzyme that allows for this process to occur. It holds these substances near one another. And then the final type, if we move this up, metal ion catalysis. We know that metal atoms, zinc, magnesium, iron, these are going to be used as cofactors by many different enzymes. We'll see as we move through our study of biochemistry that metal atoms are very important to a number of different reactions. The metal atoms lose electrons easily, therefore they can exist as cations. And that positive charge on the metal will allow the metal to stabilize transient intermediate structures. So it stabilizes intermediate structures. Again, this is the charge of that metal, that positive charge. It assists in forming strong nucleophilic species. And it holds the substrate inside the active site. So these are the 4 different types of catalytic strategies. We should be able to define these types of strategies. So now we can apply these to actual enzyme systems. And one of the first classes of substances we can look at are proteases. Now, proteases cleave proteins by hydrolysis. The overall reaction that takes place is going to be an exergonic reaction, but thermodynamically it happens very slowly. And that's because of how the peptide bond is oriented. Kind of results in a resistance of that hydrolysis. And that goes back to when we've talked about the double bond characteristic of peptide bonds in earlier chapters. Okay, so the peptide bond has kind of a built-in resistance for this, but we need to be able to break these apart. And proteases tend to function through covalent catalysis, which is based on nucleophilic attack. So to understand what takes place in the active site, we can study chymotrypsin. And chymotrypsin is a serine protease. It's found in the small intestine. It cleaves on the hydroxyl side of large bulky amino acid groups. And what it's doing is it cleaves those peptide bonds. So it's breaking up our polypeptide chain. It's the serine residue of the enzyme that acts as a nucleophile and attacks the carbon of that carbonyl. So this is nucleophilic attack here. The carboxyl side is the right-hand side. So chymotrypsin will only cleave on the right-hand side of a hydrophobic residue. So this figure is showing where we're going to end up cleaving, on the right-hand side of that carboxyl. Now chymotrypsin, as we said, utilizes covalent catalysis. This means that the active site contains a powerful nucleophile, and that nucleophile is going to form a temporary covalent bond with the peptide substrate. In this, acid-base catalysis will also be involved in the overall process of binding these things. And I think that's important because we need to know that it's not that one enzyme employs only one type of catalytic strategy. These can all work together in order to allow enzymes to function properly in harmony, really, so we can get these things efficiently working. So chymotrypsin functions through two-step catalysis. And this is known because studies have been done to probe the active site with an inhibitor. And the inhibitor that has been used in the past is this DIPF. Okay, so this is an inhibitor that you put in, and you can look at how the reaction is taking place. This is diisopropyl phosphofluoridate. So experimental data suggests that this particular enzyme is going to use a two-step process to hydrolyze the peptide bond. And to monitor the reaction, we generally use a substrate that changes color when it reacts with the active site. So you can take this information that you get, so you put this inhibitor in, we put something in that's going to produce some change that we can monitor, and then we can take that data and we can graph the data. And when we graph the data for this particular system, what you end up seeing is that there's an initial burst of product that's formed. So the color is produced pretty quickly at first, and then you have a slowing down. So that means because you have an initial really fast production of color and then a slowing down of that color, it means there are two different steps, or it suggests there are two different steps. So in the first step, we have a fast acylation step. So an acyl group is added onto the enzyme and an amide is released. And then the second step, which is the slower step, involves the hydrolysis of the intermediate complex that's formed. So this is just showing us when we have this reacting, this is a product that's formed, and we're able to monitor the presence of a colored product that's formed. This is how we do these experiments. And that colored product that forms leads to this data. This is the graph data that I was referring to. So graphing that data, you see that, again, experimental data, you have an initial burst where we're increasing the amount of product that's formed pretty quickly, and then you have a leveling off here that occurs. So that indicates that there are two separate phases or steps. And this figure is just showing that we said that the initial fast burst step was an acylation step. So we can see that here's our acylation step that forms. So you have an acyl group added to the enzyme and an amide released. Okay. And so you can see that with the addition of this substance to that particular site. And then we had that second slower step, which was going to be that hydrolysis step. And that hydrolysis step was going to be, again, much slower, but it's also a deacylation step. Now here what we're looking at is the actual structure of chymotrypsin and the active site of the chymotrypsin. So we can see that there is an environment within that active site, and there's a lot of different things that are going to take place within that, but there are some key players. We have some disulfide bonds, some serine residues that are going to be important as you move through this particular process. Now, chymotrypsin actually uses something called the catalytic triad. So the catalytic triad is a series of 3 residues. We're looking here at these 3 residues, and these are going to play an important role in working together to catalyze the cleavage of peptide bonds. So in the form it appears in the active site, we have this present as an alcohol, which is not a strong nucleophile. Okay. Histidine and aspartame are going to help convert the serine to a strong nucleophile. And I should have clarified, what I'm talking about is the alcohol, is this serine residue, which we saw on the previous slide was one of those important players within this overall structure. So serine is present as an alcohol. It's not a strong nucleophile. These other 2 residues are going to work together to help get that into a place that it's converted into a strong nucleophile. The negatively charged side chain of aspartame will interact. So we have our negatively charged side chain is going to interact with a hydrogen that's on this histidine residue here. And this positions the histidine in a proper place for the other nitrogen to interact. So it kind of holds this histidine residue in place so that this nitrogen right here can interact with the hydrogen of the serine residue. And when that happens, you're going to form this alkoxide ion. So looking at all of this in more detail, there's a multi-step process that's occurring. The reaction that shows the overall reaction mechanism of chymotrypsin. So first, the serine alkoxide, which is a modified serine residue, acts as a nucleophile and attacks the carbon of the carbonyl. So what we're looking at here, we have our start of the reaction. Here's number 1 right here. So you've got your catalytic triad, okay? We have the aspartame residue interacting with the histidine residue to hold it in place. That histidine residue is going to interact that nitrogen with this hydrogen to form that alkoxide species, so we have a nucleophile. And that's on that serine residue that's there. And it's this residue that is going to interact with the substrate, which is the thing the peptide bonds we're trying to cleave. Okay? So you end up with a nucleophilic attack on the carbonyl carbon, as we're seeing here, the carbonyl carbon of what we're trying to cleave. So this forms a relatively unstable tetrahedral intermediate. The structure is stabilized by what's referred to as the oxyanion hole, and the tetrahedral quickly collapses. Inside the pocket, there are nitrogen atoms that contain hydrogen atoms, and the hydrogen atoms will interact with the negatively charged oxygen that's in there. So that tetrahedral intermediate isn't stable for long, and it ends up collapsing. Here's our formation of the tetrahedral intermediate. Okay, so we've got our tetrahedral intermediate here, this oxyanion hole that forms, again, not stable, going to end up collapsing. When that happens, it acylates the serine and forms an amide molecule that deprotonates the histidine nitrogen. Okay, so you've got your collapse, you've got your intermediate, and here what we're seeing is that we're deprotonating. So this histidine residue here, that hydrogen that's on that nitrogen group, we're going to end up deprotonating, and we've lost it down here. In the next step, the amide is going to move away. The amide product departs, and water can come in. Okay, so the amide product is going to depart. Water is coming in here. Histidine will help transform water into hydroxide. Okay, so we have this histidine residue again. Now it's interacting with that water to help get it into a form that is a strong nucleophile, a hydroxide. Water isn't a strong enough nucleophile, so the histidine has to remove a proton to transfer water into a hydroxyl group, and the hydroxyl group can then serve as the nucleophile. And that's what we're seeing right here. We have that OH group that's forming. And this happens in a very similar way to what we saw up here in this second step. So you form that oxyanion hole again, which is that unstable, tetrahedral intermediate that collapses. This forms a carboxylic acid product. So this portion is going to collapse, and it's going to form this product. And that product is then released. So the departure of that carboxylic acid prepares the active site for another cycle so it can come in and cut another bond. So again, what we're doing is we're cleaving peptide bonds, and we're cleaving it on that carboxyl side of those bulkier or larger amino acid side groups. So just a little more, we talked about that oxyanion hole, so to look at it in a little bit more detail, really this is just a portion that comes in and helps stabilize an unstable intermediate. And again, it doesn't stay around for long. It's going to collapse, and then we're going to continue with our catalytic process. Now, the idea of this catalytic triad, it is not unique to this particular chymotrypsin substance. It happens in other catalytic enzymes as well. So chymotrypsin contains a relatively deep pocket, and that deep pocket is mostly hydrophobic. And that's going to allow for long, uncharged, and hydrophobic amino acids like methionine, phenylalanine, tyrosine, tryptophan. They'll fit down into that pocket without much electrostatic repulsion. So these particular groups are the only ones that are going to fit down into that pocket, and we refer to that pocket as the S1 pocket. So it's because of the shape of where that active site is, and again, that catalytic triad occurs within that active site. That's why chymotrypsin acts on those larger, bulkier groups, because large groups can get down in there. So the shape of that pocket gives the specificity to the chymotrypsin, even though the catalytic triad is going to be necessary in order for catalysis to occur. So chymotrypsin is typically used as the representative serine protease, but other serine proteases will exist in the body. And those other serine proteases will use the same catalytic triad. And this process involves both covalent catalysis and acid-base catalysis. And so we've seen that nucleophilic attack, we've seen that transfer of a proton within the mechanism that we just discussed. Trypsin and elastase that we see here are two additional enzymes that are going to involve this particular triad. Okay, these are also found in the digestive system. They're cleaving these peptide bonds. And the triad gives the catalytic ability. The shape gives the specificity, the shape of the pocket. So the shape of what we're looking at here, where these different residues can get down in, that is going to allow for the specificity. That triad, that group of 3 residues, is going to allow for catalysis to occur. Trypsin and elastase are going to cleave other amino acids because they have variations in the pocket that exists there. Okay, so remember, pocket gives specificity. Trypsin will cleave peptide bonds on the carboxyl side of lysine and arginine amino acids. And that's because at the bottom of the active site of trypsin, there is an aspartate residue. And the negative charge of the aspartate will stabilize amino acids that are positively charged. And lysine and arginine are positively charged. Now the elastase will cleave peptide bonds on the carboxyl side of small hydrophobic amino acids. Things like glycine, valine, alanine, leucine, isoleucine. And this is because there are 2 valine residues that are down in that pocket, and they block off a large portion of the pocket. So only small residues can get down in the pocket. So the take-home, catalytic triad is used in a variety of different processes, different proteases that are present. The catalytic activity, or the mechanism, is going to be the same. The particular position where these enzymes are going to cleave will be different based on the pocket that exists and how the pocket is modified. Now, we've been talking about serine proteases, but there are other classes of proteases as well. Not all proteases will utilize activated serine residues. So there are 3 alternatives to serine proteases that have been discovered, and these are shown here. We have cysteine proteases, we have aspartyl proteases, and metalloproteases. These substances tend to generate nucleophiles and attack on the peptide carbonyl group, but these are functioning in a similar way, except we're not using that serine residue. A different residue is going to be important. So in the cysteine proteases, here's our cysteine proteases here. A histidine residue activates a cysteine residue. So we still have that histine residue that's here, and we have a cysteine residue, and that histidine, just like it did in the other case, is going to interact with that hydrogen. So the cysteine residue plays a role in a nucleophilic attack on a peptide bond. Cysteine proteases only require histidine and not the full catalytic triad, and that's because the sulfur of the cysteine is a better nucleophile than the oxygen that we saw in serine. Aspartyl proteases, they have a pair of aspartic acid residues that work together in that pocket. They work together to allow water to attack a peptide bond. One aspartic acid residue in the deprotonated form will activate the attacking water molecule. So we have water in here, one residue is going to activate, and then that water can become a nucleophile and allow the process to occur. And the last, metalloproteases, involve an active site that contains a bound metal ion, and usually that ion is going to be zinc, as we see here. Usually it's zinc. This activates water molecule, and that water then acts as a nucleophile, and that nucleophile then attacks the carbonyl group. So in all of these cases, the whole point of the pocket or the active site for the catalytic strategy is that we have residues that are interacting with one another so that they can form nucleophiles, and the nucleophiles are going to attack the carbonyl carbon of the protein that we're trying to break up or we're trying to cut. And again, we mentioned that these were present in the digestive system. A lot of these proteases are in the digestive system because we're eating foods that contain proteins, and we need to be able to break these things down. In all three of these cases here, the active site includes features that activate water or some other nucleophile. They all polarize the peptide carbonyl group, and they all stabilize a tetrahedral intermediate, like we saw in the full mechanism for chymotrypsin. So another example we can use to look at different catalytic strategies is carbonic anhydrases. And these employ acid-base catalysis and metal ion catalysis. Again, we're not just using one type of catalysis in each enzyme. These things can work together. So carbon dioxide is a waste product of aerobic respiration. And the carbon dioxide is dumped into plasma, and it then needs to be expelled by the body. And we've talked about before how carbon dioxide is nonpolar, but the blood plasma is polar, so the CO2 can't readily dissolve. That nonpolar carbon dioxide needs to be transformed, and it's transformed into bicarbonate ion. And that bicarbonate can easily be dissolved inside the plasma and the red blood cells. We talked about this when we were talking about the Bohr effect. And that carbonic anhydrase is what's going to allow for that conversion of that CO2 to the bicarbonate. So carbonic anhydrases catalyze conversion of CO2 into the bicarbonate ions. The most widely studied carbonic anhydrase is carbonic anhydrase 2. That's found in blood cells. There are at least 7 types of these enzymes in our body. So depending on where you are in the body and where this process occurs. But remember, this is the process, if we want to relate this to something, this is the process that's responsible for that Bohr effect that we've studied. Here we're just looking at the structure of the carbonic anhydrase 2 enzyme. So you can see that the active site or that pocket in there that's going to actually aid in catalysis. In terms of active site and mechanism, carbonic anhydrases would utilize metal ion catalysis. So the active site will contain a zinc metal atom. The zinc contains a positive charge, it's +2. It's bound to the imidazole group, which is a ring structure that's part of the side chain of zinc that's attached to this, of different histine residues, as well as water that's in there. So we have histine residues and water all interacting with this zinc ion. The water is what's ultimately going to react with the CO2 to form the bicarbonate ion. And this figure is just showing that binding site so we can see the CO2 coming down into that pocket. You see your zinc, you see it interacting with these histidine residues, and you have water that's playing a role within this site. So just as we did in the case of chymotrypsin, we can talk about the mechanism of carbonic anhydrase. In this case, the zinc helps transform water into a better nucleophile, which allows it to react with the carbon dioxide at a higher rate. And that allows us to form bicarbonate at a higher rate. So based on experimental data, the binding of the zinc atom to water will end up lowering the pKa value, and this makes water much more likely to give up its hydrogen ion, and it becomes a hydroxide ion. And that hydroxide can serve as a nucleophile, so it can participate in nucleophilic attack. So zinc generates hydroxide by facilitating the release of H+ from water. So we're taking that water and we're producing that hydroxyl group so we can form a nucleophile. Carbon dioxide binds to the active site and is positioned to react with that hydroxyl group. The hydroxyl group attacks the carbon dioxide, and that results in that bicarbonate production. And then the active site is regenerated with the release of this bicarbonate ion. So we can look at this process pictorially here. You have water binding to the metal zinc atom here, substance. There's our histidine residues and our zinc, and water comes in and binds to this. Partially negative charge of oxygen is attracted to the positive charge of the zinc. Okay. When that interaction occurs, the hydrogen, one of the hydrogens of water, is much more easily removed, so it's able to dissociate, and we see that happening here. That's going to lead to the formation of that hydroxyl group. Okay, so that takes this water, which will not serve as a nucleophile, and converts it into something that serves as a nucleophile. At this point, carbon dioxide is going to come in. We have that nucleophile there, which can attack that carbon dioxide. We see that in this process. That forms this bicarbonate intermediate. We can see in this step here. Once the intermediate is formed, then you have another water molecule that comes in, and that water molecule that comes in and kicks this piece out. So we can regenerate the initial substance here. And then, of course, that bicarbonate ion is going to be transported by the blood plasma and participate in that change in pH that we've seen with the Bohr effect. Now, there are a couple of slides in here that I'm skipping over. So if you look at what the actual PowerPoint presentation shows, a couple of extra slides. But the take home from these slides that I'm skimming over is that under certain pKa and pH values, a proton shuttle system is established, and that allows buffer components to participate in a reaction because this is going to impact the rates of the overall reaction. Okay, but we're not too worried about the details of these couple of slides that I'm not specifically going to review. So a third type of enzyme we said we were going to look at were restriction endonucleases. Okay, these recognize particular base sequences. There's a recognition site in target DNA, and it cleaves that. So restriction enzymes. We know that restriction enzymes will show a tremendous amount of specificity. Okay, they must not degrade host DNA, which contains the recognition sequence. They want to cleave only DNA molecules that contain recognition sites without cleaving any other molecules that lack these sites. These are also going to occur using nucleophilic attack, but it does so at the phosphorus atoms of the sugar phosphate backbone. Okay, so we're going to look at some of these here. We see this, there's our sugar phosphate backbone right here. We have water coming in and playing a role, and we're going to go through and talk about this mechanism. So there are actually two different mechanisms by which the restriction endonucleases can function. In mechanism 1, it utilizes formation of a covalent intermediate. So that's this one right here. And mechanism 2 involves direct hydrolysis of that particular peptide bond, or not peptide bond, rather, that particular portion of the sugar phosphate backbone. This is just showing us that specificity. There are specific places where different restriction endonucleases are going to come in and recognize and cleave. So it's going to cleave the actual sugar phosphate backbone there. In general, restriction enzymes will involve metal ion catalysis. They require magnesium ions in order to function. Okay, so we see this magnesium ion present here when we're looking at the overall structure of this particular enzyme. And then that other class of enzymes we said we were going to look at were myosins. And myosins are enzymes which catalyze hydrolysis of adenosine triphosphates, ATP, to form ADP and inorganic phosphate. They also use the energy that's associated with this process, which is thermodynamically favorable, to drive the motion of molecules within cells. So here what we're looking at is our ATP and water playing a role, and that water is going to aid in breaking apart or removing a phosphate from the overall structure, and then forming ADP, well, which is what's left after that phosphate group comes off. This is showing the complex. We have our myosin, our enzyme there, and that magnesium plays a role in that pocket with that enzyme. Our ATP will come in. We'll be able to, because of the interaction that's taking place within that pocket, cleave off one of those phosphate groups and form that ADP. So here, this is just kind of showing what's going on with that ATP structure. Again, we're interacting with water. That's going to play a role here. We do have that metal ion catalysis involved to help create that intermediate that we have. This is going to allow us to cleave this particular phosphate group off, and our ADP is going to be left. And on that last slide, you saw those dotted blue lines. That's just indicating that we're forming some transition state or some intermediate between the water and our overall ATP structure. Just like the cases of the other enzymes we've looked at, myosin will essentially use acid-base catalysis. The ATP serves as a base. It promotes its own hydrolysis in this case. But this reaction, it uses the acid-base catalysis, but we've also seen, because magnesium is important in this process and is in that particular active site, it will involve metal ion catalysis as well. Now, there are a few more slides present in the chapter presentation, but I think this is a good bit of information. And so kind of the take home for chapter 9, what you want to be comfortable with, you want to be able to describe those different types of catalytic strategies. So we outlined each: covalent, general acid-base, catalysis by approximation, and metal ion catalysis. And then we looked at how those were involved in specific examples of enzymes: serine proteases, carbonic anhydrases, restriction endonucleases, and myosins. So describe the types, list and describe the types of catalytic strategies, and then list and describe classes of enzymes that utilize these catalytic strategies. What's the general mechanism of these enzymes? What types of catalysis are involved in each of these enzymes?